Hello everyone. Today we're going to talk about experiment number 4, which is titled The Effective Temperature, pH, and Detergent on the Cell Membrane. So we all know what temperature is. pH is basically the amount of acidity or alkalinity the solution has. So basically the amount of hydrogen ions in a solution. Detergent indicates chemicals that are found in cleaning products such as Tide, such as Fairy, etc. Okay, these chemicals, they help solubilize fats, okay, so that we can wash them away. Okay, so this is what detergent is.
Now, I always like to present the big idea to relate it to the medical field. So the big idea for this lesson is to understand diseases that occur due to disruptions in the plasma membrane. One of those diseases is cystic fibrosis. And in cystic fibrosis, what you have is basically a transmembrane protein called CFTR that has been mutated. So, in the normal case of CFTR, in the normal case of cystic fibrosis, in the lungs, it functions as a chloride transporter. Transports chloride ions back and forth across the membrane. However, what happens in cystic fibrosis is that there is a mutation in this CFTR gene. Okay? And because of this mutation, this deletion actually, this transmembrane protein, as you guys can see from the picture, basically does not function anymore. Okay? So it destroys the 3D structure of the protein. And because of this change, okay, patients with cystic fibrosis, as you guys can see from the CT scan here, they develop thick mucus in their lungs. And according here to the picture with the symptoms, all of this leads to, for example, sinus problems, trouble breathing, etc. So you can see here, guys, that because of a change in the 3D structure of a transmembrane protein, in this case, because of a mutation that is inherited from your parents, for example, okay, this changes the 3D shape of the protein, and this leads to disease. So that is why we're going over this experiment, because a change in the transmembrane protein, in the 3D shape of a transmembrane protein, which is of course present on the plasma membrane, changes its 3D shape and can lead to several diseases, and one of these diseases is cystic fibrosis. Alright, so we have 3 objectives for today. The first objective is to briefly review the structure and function of the human cell membrane. So we can say cell membrane or plasma membrane. They are synonymous. The second objective is to determine the effect of pH and temperature on the cell membrane integrity. The third objective for today is to determine the effect of detergents on cell membrane integrity. So all of these factors, pH, detergent, and temperature, these are all external factors. We will see how these factors can disrupt the plasma membrane. Now, as you guys might know from the theory, the plasma membrane is the boundary that separates the living cell from its surroundings. If you guys think of Saudi Arabia, as a big country, it has boundary with certain countries. It's selectively permeable, okay, which means that only certain people can come in and certain people can come out. Okay? If we are not selective, then Saudi Arabia would lose its identity. A pancreatic cell would be identical to, for example, a liver cell, a hepatic cell, or would be identical to a skin cell. So the cell must show selective permeability, meaning that it allows certain molecules in selectively. Okay, for example, oxygen can diffuse in briefly because it's a small molecule and it's nonpolar. Whereas glucose, it's polar and it's a large molecule, so it needs carrier proteins to transport it in. That is selective permeability. Now, the plasma membrane is composed mostly of phospholipids. Okay, but it also has proteins. Okay, it also has proteins and carbohydrates. When we say that the plasma membrane is a mosaic, here we're talking about it's a mosaic. It has a mosaic, different kinds of proteins, as you guys will see in the next slide. Okay, so phospholipids are amphipathic molecules. Amphi in English means both. Pathic means condition, okay, a certain condition. So it has two certain conditions. In this case, it has a hydrophilic head, which faces the water. Okay, the water can be on the outside of the cell or the inside of the cell. Here we're talking about the cytoplasm or the external environment. The hydrophobic tail, okay, the two tails of the phospholipids, they face each other. Okay, because they're made of hydrocarbons, they are pretty hydrophobic, and they face each other. They are afraid of water, hydrophobic. So as you guys can see from this slide, that the plasma membrane is actually a fluid mosaic. So it's not completely solid, it's a fluid because the phospholipids can move laterally. Okay, they can move laterally, and if you look at YouTube videos, you will see the phospholipids move. Okay, so it's sort of like jello-like structure, jello. It's a mosaic because it's a mosaic, it's a mix of proteins. As you guys can see here, we have glycoproteins. Okay, here where the proteins, they have a carbohydrate attached. We have peripheral proteins, we have integral proteins, and all of these can function, they have different functions. So that is what is meant here by a mosaic. Now, let's talk, guys, about molecule movement in cells. There are two types of movements of molecules that occur in cells. These are passive transport and active transport. In passive transport, there is no energy required. In active transport, energy is required. And its movement, basically, passive transport, its movement due to a gradient. Now, this gradient can be concentration, can be pressure, can be charge, so it doesn't have to be concentration. And in passive transport, as we said, this doesn't require energy, and its movement from high concentration of solute, high concentration of glucose, for example, to low concentration to make the concentrations equal. Active transport, which requires energy, it is the opposite. So it is movement from low to high concentration. That is, against their concentration gradient. When we talk about passive transport, we don't say against their concentration gradient, we say down their concentration gradient. Yep. Now, guys, there are three types of passive transport. So we are focusing here on passive transport, which doesn't require energy. So the first one is diffusion, and that is the movement of molecules from a high concentration to a low concentration. This doesn't require energy. Think of perfume or cologne. When you spray perfume or a spray in the room, basically the perfume moves from high concentration and so it spreads to low concentration of the room. This is the natural way, and there is no energy required. This is diffusion. The second type is osmosis. Osmosis is basically the diffusion, so we're still talking about diffusion, but in this case, it's a name that is dedicated to water. So it is the diffusion of water across a selectively permeable membrane. And in this case, so focus with me here, guys, this can tend to be a bit confusing. So here we're talking about when water, so basically osmosis is when water moves from low to higher solute concentration. Notice here, guys, the word solute. Okay, so we're not, of course, water is not the solute, water is a solvent. So water moves from low to high solute concentration. This solute can be sugar, for example, to equalize the concentrations. Okay, and this often involves movement of water into the cell or out of the cell. So in both can happen in both directions. Now, the third type of passive transport is facilitated diffusion. And in this type, the molecules, they require help. Okay, they require help. These molecules, for example, they are polar, they have a charge, they require help to enter or leave the cell membrane. So facilitated diffusion requires transport proteins which speed up the passive movement of molecules across the plasma membrane, And there are two types, the channel proteins, and as you guys can see from the picture, it moves molecules from high concentration to low concentration. It's a channel, like a tunnel. The second type is they are carrier proteins. They carry, okay? Like when you carry somebody on your back, for example, and these carrier proteins, obviously, they carry molecules from high concentration to low concentration. Okay? So, of course, this doesn't require energy. Now, please guys, focus with me on this slide. This tends to be a bit confusing for some students. So, we're looking here just at the membrane on the left side. Okay, on the left side. So, the membrane or the tube on the left side, that looks like the U shape, of course. This membrane, you can see here the yellow dots. Okay, it's permeable to both the solute and water. Okay, so the solute will move by diffusion, the water will move by osmosis. Now, eventually our goal is to make the concentrations equal on both sides. So the solute will move from higher concentration to low concentration. Okay, because it can pass through. So if it moves from high concentration to low concentrations, okay, this will be equal. The concentrations will be equal, and you know, if you guys remember, by osmosis requires water to move from low solute concentration to higher solute concentration. In this case, both concentrations are equal, right? Okay, because the solute has already moved by diffusion. So you can see here in the arrow that the volume will be the same on both sides, and we have equal concentrations. In the other case, on the right side, here the membrane is only permeable to water. It's impermeable to the solute. So the molecules will not move. Only the water will move by osmosis, and it will move to the right side. Why to the right side? Because it moves from low concentration of the solute, of the red dots, to high concentration. Okay? So on the far right side, this is the end result where you have the same concentration, the same osmolarity, but of course you can see here the volume is more. There's an increased volume on the right side. We have the same concentration, okay, we have the same concentration but different volume. Okay, because water has moved by osmosis from the left side to the right side. Now, as you guys might already know, all proteins, they have a 3D shape. They have a tertiary structure that determines their functions. And you guys know from the theory class how 3D structures form due to the interactions between the R groups. And of course, there are different types of interactions. Now, we can destroy these 3D structures, for example, by mutations, as you guys saw here with the cystic fibrosis in the second slide, I believe. But we can also destroy them by external factors, such as temperature, high temperatures, pH. Okay, and you can see here from the picture that the protein, the enzyme, let's say this is an enzyme that carries, for example, glucose. It has a nice 3D shape, specific, for example, to the glucose shape. If I destroy its 3D shape, okay, meaning I denature this protein, I unfold it, I destroy its function. And if I destroy its function, okay, it will not perform its regular normal function in the cell anymore. Okay, for example, if it transports molecules in and out, stuff will leak. Stuff will leak outside the cell, and stuff from the outside will come to the inside. So by destroying the 3D shape of the protein, such as by pH, temperature, detergent, we denature the protein. We move it away from its nature, okay, from its natural form. So, as this slide indicates, in order for a living cell to survive, to perform its regular function, its specialized function, it must have a functioning, selectively permeable membrane. For a pancreatic cell to be a pancreatic cell, it must be selective, and who comes in, or what molecules come in and what molecules come out. If the proteins, okay, which are part of the mosaic, or the phospholipids are distorted or destroyed or malfunction, if they malfunction, this can affect the permeability of the membrane, and leakage can happen, okay, as you guys will see from the experiment.
So what happens when you add detergent to the plasma membrane? When you add detergent to the plasma membrane, which consists of course of the lipid bilayer, you form what's called a micelle. Now, detergent molecules, as you guys can see from the picture, are very similar to phospholipid molecules. A detergent molecule has a hydrophilic head indicated in red, and instead of two hydrophobic tails, it has one hydrophobic tail. So here you can see a structure of one type of detergent called SDS. It has a charged hydrophilic head, and it has a long hydrophobic tail. Now, when you mix this with the plasma, with the bilayer, with the phospholipid bilayer, you have what's called a mixed micelle. And a mixed micelle is not the same as a bilayer, so it's a monolayer. It has one layer, and this creates basically a hole in the cell membrane. It makes it very permeable, and things can leak out easily. So it basically destroys the plasma membrane because it's not selective anymore.
Alright guys, so solution differences in cells. I will go to the next slide, and when I go to the next slide, this will help you guys understand this slide. Okay, because these two slides, this one and the next one, they are this about the same. They talk about the same thing. So I will move on to the next slide. Alright, solution differences surrounding red blood cells. So imagine you have a red blood cell, one RBC, one erythrocyte, one red blood cell. You put it in a solution. Now, if this solution has the same concentration of solute as inside the RBC, so let's say the red blood cell RBC has the same concentration of solute as the outside solution, we call this isotonic. Okay, isotonic solution. Okay, and this is what happens when you will see guys see later on when you put red blood cells in a solution called PBS. PBS. Okay, because PBS is a solution that has the same concentration of solute as inside red blood cells. Here, the cells in an isotonic solution, they are pretty happy. Now, if I put them in a hypertonic solution, so think of the word guys, hypertonic. Hyper means increase, enhanced, lots of. So if I put them in a hypertonic solution, meaning the solution outside the red blood cells, the outside, has a higher concentration of solute. Now in this case, if you guys remember from the previous slide, water will move from the low concentration of solute to the high concentration. So in this case, the high concentration is on the outside. So water will move from inside the red blood cell to the outside. Inside, from the red blood cell to the outside, into the solution. And this will cause the red blood cell to shrink, okay, to shrivel. This is called crenation. Crenation. Now on the opposite end, on the right side, hypotonic solution. In a hypotonic solution, you have a lower or a decreased amount of salt or solute compared to the red blood cell. In this case, the opposite will happen. The opposite will happen. Water will move from the outside of the cell to the inside of the cell. Okay, by osmosis. So all here we're talking about here, guys, osmosis, both in the case of hypertonic and hypotonic. So in hypotonic, water will move from the outside to the inside, and you guys can guess what will happen. This will lyse the cell. This will rupture the cell, or this means this will break the cell open, and the cell content will be released into the solution. Okay, and one of this cell content is the hemoglobin. Alright guys, so for today, for experiment number 4, we're going to test all of these external factors. The 3 external factors: temperature and pH and detergent. We will see what their effect will be on the cell membrane. Okay? Will they rupture the cell membrane? Will they leave it intact? We will see today. Now, to test all of these effects, to test the effect of temperature, pH, and detergent, we will use red blood cells as the model. So another name for red blood cells is erythrocytes. And we will use human peripheral blood. So here when we're talking about peripheral blood, we mean blood that is circulating in our system, not the ones present, not the blood present in our lymphatic system or the spleen, etc. And you guys can see that if we rupture, if we break the cell membrane, hemoglobin will be released. So it will be a visual observation. It will be a visual observation to see the effect of these external factors on the plasma membrane. Now, let's talk about hemolysis. So we're actually going to be looking for hemolysis. Hemolysis means, hemo means blood. Lysis means breaking. So hemolysis, hemolysis. So we're going to be looking for hemolysis, which is the breakage of red blood cells causing the release of hemoglobin and other internal components. If you look at test tube A and B, guys, look at test tube A and B. Here we have no hemolysis. This is the regular color of the red blood cells, which is opaque red. Opaque means cloudy. So you can see A is pretty cloudy, opaque color. B, also no hemolysis. It's basically after leaving the test tube 60 minutes or 1 hour, on the bench. You can see that the red blood cells precipitate and you have the plasma on top. You still have that cloudy red color. Sometimes it's pink, but it's cloudy. So if you see the cloudy color, cloudy pink or cloudy red, this indicates that there is no hemolysis. Now, if there's hemolysis, if you add a hemolysin, as you guys can see in C, hemolysin basically means a toxin that is released by bacteria, because some toxins can act to break red blood cells. If this happens, you have hemolysis. And according, you guys can see here, test tube C, if hemolysis happens, you have a clear red color. It loses its opaqueness, there's no more cloudy color, and you can see through. So you have a clear red color, transparent color, indicating that hemolysis has occurred, has happened. So this is a result that you guys might see. Okay, so on the far left side, the test tube on the far left side, it has an opaque color, an opaque red color. This indicates that there is no hemolysis. That's why you see here the negative sign. In the middle, you have one positive, you have a clear red. This indicates that hemolysis has occurred. It's not cloudy, it's not opaque anymore. On the far right, okay, here you have lots of hemolysis. Lots of hemolysis. And when you have lots of hemolysis, when you add, for example, a strong detergent or very acidic solution or very basic solution, the color turns into clear brown, clear black, etc. So it's still clear, but it has a darker color, indicating that lots of hemolysis has occurred. Alright guys, so the first thing we're going to do is we're going to test the effective temperature on the cell membrane. Okay, so let's move on to the next slide. So this is just the materials that we need.
Alright guys, so this is the procedure for the effective temperature on the cell membrane. The first step is to label 3 test tubes and add the following reagents here in the table. For all tubes 1 to 3, these tubes correspond to the different temperatures, you add 1 ml of PBS. PBS solution is basically a solution where the cells are in a nice environment, they are in an isotonic environment. And then we add 100 microliter of blood sample. In step number 2, we gently vortex. Vortex is a machine that I would have showed you guys how to use, but it's basically, it mixes the solutions to make them homogeneous. And then you incubate at the different temperatures. So then you incubate at test tube number 1 at the normal body temperature of 37 degrees, 37 Celsius. At number 2 at 60 Celsius, which is an extreme temperature. So what do you guys think? Is there going to be hemolysis or no hemolysis? And number 3, test tube number 3 at minus 80, which is way below 0. So cells don't like this environment. Okay, and then we're going to incubate for 5 minutes. We're going to incubate for 5 minutes, and then we're going to observe our results. Okay, then you're going to observe and record your observations in table A of the question sheet. So have a look at your table A of the question sheet. Okay, and the observations are there.
Alright guys, so what do we expect to see? what is the effective temperature on the cell membrane. At extremely high, such as the 60 Celsius, or low temperatures, such as the minus 80, we expect to see hemolysis. At very high temperatures, as you guys have learned earlier, that proteins become denatured. They lose their 3D shapes, they lose their overall structure, their three-dimensional structure, okay, and they lose their function. So you see that hemoglobin will get released. This creates holes in the cell membrane and hemoglobin will get released. At very low temperatures, such as minus 80, what happens here, guys, that when water freezes, it actually expands. And by expanding, it pushes against the cell membrane, and this causes it to rupture or break. So we also expect here hemolysis to occur. At body temperature, which is perfect temperature for the red blood cells, we expect no hemolysis, of course. So in question 1A, guys, it talks about, based on your findings above, are your results expected as expected? Slide 23. This is slide 23. So this is what we expect. Now, the second external factor that we'll be testing is the pH. What is the effect of adding extreme pH, okay, on the basic side and on the acidic side to the cell membrane? So the materials that we need, of course, is the human peripheral blood, dilute acidic solutions such as HCl, which has a very low pH, dilute alkaline solution such as sodium hydroxide, which has a very high pH, very basic, and PPS, which is the perfect solution for red blood cells. It's isotonic and it's at neutral. It has a neutral pH, pH 7.4. Let's find out. Now, the procedure. The first thing we need to do is to label 3 test tubes and add the following reagents. So for test tube number 1, this will be the neutral. This will be under the neutral condition. We are putting the red blood cells, the erythrocytes, under the neutral condition. 1 ml of PPS with 100 microliter of blood. Test tube number 2, this is the highly acidic solution. So we're adding HCl, hydrogen chloride, okay, 1 ml of hydrogen chloride, HCl, to 100 microliter of blood. And test tube number 3, 1 ml of the base, the strong base, sodium hydroxide, we're adding to it 100 microliter of blood. Step number 2, we are vortexing to mix the samples. Step number 3, we're incubating the sample for 2 minutes, letting it stand for 2 minutes. And then step number 4, we're going to record our observations in table B. Okay, so you will see my observations in table B. Let's find out, what do we expect? So what do we expect, guys, for pH? So acidic or basic pH, basically what it does is that it disturbs the charges on the membranes. Okay, it disturbs the charges on the membrane components, leading to hemolysis. For example, one of these membrane components are proteins. And proteins, they form their tertiary structure, their secondary structure, and their quaternary structure. If I add an extremely acidic pH or an extremely basic pH, this interferes with the charges. And when the charges are affected, this creates a change, an alteration in the 3D shape of the protein, therefore destroying its function and creating holes where now this causes hemolysis. So basically this allows for the red blood cells to leak to the outside.
So next, guys, we're going to test the effect of detergent on the cell membrane. But we're also here going to test the effect of putting the red blood cells in a hypotonic environments. Okay, so these are the materials that we need. We need human peripheral blood, okay, whole blood. We need 2 detergents. We need to test 2 detergents, saponin and Triton X100. However, we're going to add SDS instead of saponin, okay, because we don't have saponin in the lab, so we're going to substitute that with SDS, which is a strong detergent that is commonly used in labs, okay, to break down cells, okay, open up cells so we can study proteins through research. So the 2 detergents that we'll be using are SDS and Triton X100. We're not going to be using saponin. Okay. We're also going to be putting red blood cells in PPS, which stands for phosphate buffered saline. Okay, this is a great environment for the cells. It's the control. pH 7.4, it puts the cells in an isotonic environment. No detergent here. This is the control. And to put the cells in a hypotonic environment, we're just going to put them in distilled water, in just water, and see what happens.
Alright guys, so this is the procedure for the effective detergent on the cell membrane. Step number 1. You're going to label 4 test tubes with the reagents, with the following reagents according to the table here. Test tube number 1, this will be your control, which is just PBS, isotonic solution, no detergent. Okay, and then you add to all the test tubes 100 microliter, of course. Test tube number 2, you're testing the first detergent, which is Triton X100. Test tube number 3, you're testing this strong detergent SDS, which we substituted for saponin. Okay. Test tube number 4 is just water. We will be putting red blood cells in water, and this is a hypotonic solution, not good for water. Step number 2, we will vortex. We will mix the samples. So again, the vortex is a machine that will mix the sample. Step number 3, we'll let it wait or incubate for 2 minutes. Step number 4, we will record our results in table C. And you guys will see my results in table C. Alright guys, so what do we expect to see when we add detergent to the cell membrane? Well, as I explained earlier, what happens when you add detergent to the cell membrane, detergents, they have one leg. Okay, they have a hydrophilic head, like the phospholipid, but they have one hydrophobic tail. So when you add when you add the detergent, whether it's Triton, whether it's SDS, this interferes with the phospholipid bilayer and creates a micelle. Which is not good for the cell. Okay, they permeabilize the cell membrane, so things will leak out, molecules will leak out. Okay, for example, hemoglobin will leak out, and hemolysis will occur. Okay, because they interfere with the hydrophilic interaction. The micelle is it's a monolayer, and it's not good for the cell. Okay. Alright guys, so again, Triton and SDS, here in the slide also says saponin. So Triton, SDS, saponin, they're all detergents. And they affect the phospholipid bilayer. How do they affect it? As mentioned in the last slide, they affect it by creating this micelle, okay, this micelle structure, which is not very good for the cell, and this we expect to see hemolysis. Now, what do we expect to see when we put cells in a hypotonic environment in just water? This causes water molecules to enter very quickly across the cell membrane by a process that I mentioned earlier, which is osmosis. And water will rush inside, and this will cause lysis of the cell. Okay, it will cause lysis of the cell membrane, will cause it to be broken, to rupture, and hemoglobin will be released. Okay, so a hypotonic environment is not good for the cell. Now, I want you guys to look at the results for the temperature part, which is table A. So this is what I got when I did the experiment. Okay, so at 37 Celsius, which is the normal body temperature, I got opaque red. Okay, and you can see the slide for this color. At 60 Celsius, I got clear red. At minus 80, I also got opaque red. Okay, so if you look at table A in the worksheet, in the lab questions, I have these observations in the table. Okay, so I want you guys, okay, to look at this table A and write the results, fill in the rest of the table, and write your explanation. So fill in the blank, question 1A. I will post the answers later on. Now, table B is the effect of pH on the cell membrane. In a neutral solution, pH 7.4, PBS, I got opaque red. Okay. In the highly acidic solution, HCl, I got clear red-brown. And in the basic solution, the alkaline solution, I also got clear red-brown. So in the worksheet, I want you to look at table B, please. Fill in the rest of the table and answer B1. Again, I will post the answers later on. Alright, Finally here, guys, are the results for the detergent part, the detergent effect, and the putting the cells in a hypotonic solution. So in PBS, which is the control, pH 7.4, no detergent, isotonic solution, I got opaque red. Triton, I got clear red. SDS, I got clear red and brownish. So clear red and brown. And water, which is putting the cells in a hypotonic solution, I got clear red. So again, guys, fill in the rest. Go to the worksheet, lab report. Fill in the remaining table C, the remaining part of table C, and answer C1.